Welcome you all to the next class on Inorganic Chemistry of Life, Principles and Perspectives. In the previous class, we have started looking at the role of vanadium in biological systems. We have tried to look at among the various oxidation states of plus and minus of the vanadium that is possible in chemical species. The most prominent and prevalent oxidation states in the biological context are +5, +4 and +3. We have looked for the reasons for these ones, because the potential reduction potential from +5 to +4 is quite large positive, and the +4 to +3 is smaller but positive, and beyond that when you go to the +3 to +2 it becomes a negative potential, and the vanadium 2 plus is a strongly reducing agent. So therefore, vanadium 2 plus is being not identified in the biological systems at all. So that is what we have noticed. We also try to understand the vanadium presence in the form of vanadates. Multiple kinds of vanadates, mono, di, tri, tetra, etc., which is dependent on both the concentration as well as the pH, because the pH will change the protonation deprotonation states of these species. Then we have looked at the vanadium 51 NMR, how that is relevant to the coordination sphere, and the basically dependent on the summation of the electronegativities of the primary coordination species to the metal center. And it is very nice that when such a vanadium 5 is present in the proteins or biological enzymes, one can find the coordination sphere quite nicely by using that. On the other hand, if you have a vanadium in the form of +4, you can use EPR as the useful tool, because the vanadium 4 plus is D1 system. So in fact, also shown in the previous class that the vanadium 5 that is entered into the biological cells can get reduced to 4, and in some cases even 3. Therefore, in biology you would see as +5, +4 and +3. And we have seen a special case of vanadate which is acting as a inhibitor for the ATPase. The ATPase goes through the two parts of the cycles. The first cycle where the sodium binding followed by release of this, then the second cycle potassium binding followed by the release of this, and the first one is driven by the phosphorylation, and second part is driven by the dephosphorylation. At the second stage where the dephosphorylation takes place, if the vanadate is present, vanadate binds to the protein much more strongly than that of the phosphate, and it is irreversible. Hence, the vanadate acts as a inhibitor for the ATP cycle. Now, let us in this class let us look at a positive role of the vanadium, that is the vanadium enzyme. So, one famous example is the haloperoxidases. So, vanadium dependent haloperoxidases. So, haloperoxidases are quite popularly known, particularly from their presence in the sea algae, where a lot of organic moieties in the ocean are converted to the halogenated ones. This could be chloro, this could be bromo, this could be iodo. So, therefore, chloroperoxidase, bromoperoxidase and iodoperoxidase. So, these are general class of the haloperoxidases, and these haloperoxidases work in the presence of the halogen, of course, is required, and what is required is a hydrogen peroxide. So, one can read this particular slide to get the most background information about this. Let us look at one example of this vanadium bromoperoxidase. We will come to the reaction etc. a bit later, and you see a huge protein. Of course, this is one monomer of the entire structure, and you can see the structure is having the alpha helices, which are mostly in the red in color, and the beta sheets which are in the green in color, and then you have the random coil which are in the yellow color. So, this whole thing together you have a 3-dimensional structure, and such a 3-dimensional structure would basically is responsible for the activity of the enzyme. Now, how is the vanadium is placed? So, let us look at to understand this, let us look at this slide. and in this slide there are two structures, one on the left side, one on the right side. Okay, so the left side one is taken from the vanadium bromoperoxidase. V-BPO means vanadium bromoperoxidase, V-CPO means vanadium chloroperoxidase. So, bromoperoxidase and chloroperoxidase that you have, this is the enzyme structure, not the entire enzyme structure is shown, only the structure very close to the vanadium part. So, how is the vanadium is placed? Vanadium is here in the center and the 4 oxo groups are there. So, it is basically vanadate is suspended in the protein where the only one direct covalent connection between the vanadium and the protein is this particular thing, which is a histidine. So, vanadium to N or the histidine is only one single connection that you have between the vanadate and the enzyme, and as the oxygens present on the vanadate are indeed involved in the hydrogen bonding, and this is from bromoperoxidase. The complete hydrogen bonding scheme is not shown here. However, if you look at another case, chloroperoxidase, we can just see only the vanadate. You see that 1, 2, 3 and 4 kind of oxygens being present here, and these are connected by the lot of hydrogen bonding situation. So, it looks like as if a vanadate species is suspended into a protein, and wherein it is attached through a one covalent connectivity with the nitrogen or the imidazole or the histidine moiety. So, you can see that all these arginines, this arginine and so other serine and of course the histidine, and this histidine is a covalently bond. So, this is how roughly the active center of the vanadium looks like in haloperoxidases. And as you see that this whole species is stabilized by all this, therefore, these you call it as a secondary interactions, and these hydrogen bonds with the side chains of the protein is called basically the secondary interactions, whereas this is the primary coordination sphere. I think this much of information about how the vanadium center is present is more than sufficient enough to understand. Of course, the rest of the protein is always there, which is not shown over here. Okay. And let us see what kind of reactivity and how do one measure such a kind of a reactivity. Haloperoxidase activity is basically it does halogenation of the substrate. We have given here case for how to assay such a kind of a the halogenation. So, this is the substrate we have, and the substrate has two forms which are shown, and this has an absorption spectrum around 290 nanometers, 20,000 mole inverse. This is 20,000 mole inverse is the epsilon value. Now, this when you add the vanadium bromoperoxidase VBRPO in presence of the H2O2 and bromide, is of course inorganic bromide like sodium bromide, ammonium tetraalkyl ammonium bromide, any of these things can be taken, and that bromo will be added here, and then what will happen is the absorption maximum will not shift, but the absorption epsilon, that is the extent to which it absorbs, is diminished. It is going from 20,000 to 100. So, a lot decrease in these ones. You can also use other kinds of substrates like a phenol red you have, and the phenol red will become the phenol blue here. This goes from 433 nanometers to 593 nanometer, which is almost 160 nanometer shift. So, you can find either by the intensity variation or by the epsilon maximum position whether the enzyme activity, and this is what is referred to as the assay method. So, if you want to identify the enzyme activity, always you require assay method, and this is one of the assay methods or actually two different assay methods for the haloperoxidase system that you have. Okay, on the next slide I have tried to put all the possible steps that one can visualize when the haloperoxidase act on the substrate. If you recall that one other thing I have forgotten to tell you is in all these things the vanadium is in vanadium 5 oxidation state. Okay. So, therefore, in the enzyme when we looked at here this particular part of the enzyme. This is basically vanadium is in the vanadium 5. Okay, now let us look at the mechanistic aspects of this particular enzyme, chloroperoxidase, bromoperoxidase, it does not matter. One of these we can, a general mechanism or a generic mechanism for this that we can understand. If you recall, this enzyme functions, one you need H2O2, other you need halide. There are two components which are required in addition of course to the substrate and in addition of course to the enzyme. So, the enzyme does catalytically, there is no question about it. So, this can be grossly looked at some kind of a steps. So, we can say the step 1 is the one where the H2O2 is being added, okay, and then H2O2 is added, obviously will add a protonation as well as attack at the metal center. So, thereby form some kind of a peroxo kind of a compound. And such a peroxide compound in the third step will activate the halide to form a reactive halide species. And such a reactive halide species very quickly dissociates by adding the halide species to the substrate and returns back to the normal part of it. So far, the direct evidence has not been seen for the any halide binding to the vanadium, direct binding of this, and still the hypohalide kind of a species is still being visualized. And the second aspect is the during the catalysis there is no change in the oxidation state. The oxidation state remains to be the +5. So, therefore, these two are absolutely important aspects to keep in mind while building the mechanistic aspects. So, as you can see from here that this is the vanadate species which is bound to the protein through this particular histidine, and this is activated by a different histidine, etc., etc. And this particular center of the enzyme is attacked by the hydrogen peroxide, and that will add a protonation to this. So, this will protonate this one, and HO2 minus is obviously a kind of a nucleophile which will act at the metal center. Okay, so, that will basically give a species of this kind of a peroxo species. Now, at this stage I should take a little bit of deviation and explain you some things that are noted. In fact, in the literature, the small molecular components that people have prepared, they have looked at the addition of the for the vanadium 5 compounds, they have looked at the addition of the hydrogen peroxide, and peroxo bound species were isolated. So, therefore, there is sufficient amount of proof evidence or support for the bonding of this peroxo species to the vanadium center. So, such kind of complexes are very well known in the literature from the coordination chemistry point of view. So, therefore, such a binding is very well visualized. Now, at this stage of the thing, the halide activation takes place. So, halide activation goes with an attack on this peroxo species, sort of break this peroxo species to form a species of the hypochloric acid. If it is a bromine case, hypobromic acid, hypoiodic acid. So, hypohalide kind of an intermediate, and certainly this intermediate is not a very stable one and very reactive. So, at this stage it will re-stabilize the coordination at this, the water as a coordination, and then that will be setting with the histidine. So, in other words, this particular species, when you have a substrate here and substrate gets halogenated. So, the product will be halogenated product and brings back this. Since the substrate is not shown over here, so therefore, it is shown as if it is the HOCl that is coming into this. Okay. So, the main mechanism is that you have the vanadium 5 vanadate based species, the H2O2 reacting to this, adding protonation and HO2 minus nucleophilic attack, and a kind of a peroxo species for which there is a lot of evidence from the small molecular chemistry, peroxo bridging, peroxo binding, and this is being further activated by the halide species to result in the hypohalous acid. And if you have a substrate at this stage, the substrate will get halogenated. If the substrate is not there, you can expect as a HOCl being liberated. Okay? And this is how one can try to understand the mechanism of the peroxidase. So, let us look at another evidence that this is indeed is the reaction center. How do we know this is the actually the reaction site or active site? So, in that case what one needs to do? Now, remove that vanadate part. How will you remove? It is very simple. You can remove the whole vanadate. So, you remove, then once when you remove this, what is the kind of a protein that you get? The protein you get when you remove the vanadate is called the apoprotein. So, therefore, apoperoxidase you will get. So, now there is no vanadate species. To this you can add sulphate. In fact, sulphate ion SO4 minus, HbO4 2 minus is very similar, and they go and fit into the almost the same region where the vanadate was there and the sulphate goes. Now, once the sulphate goes, as you can see here, the sulphate gets adjusted to the coordination or through the interactions with the protein, and now no more reaction is taking place. So, this is exactly the vanadate species which was necessary for the reactivity. So, this will reconfirm that the vanadate center is indeed responsible for the reaction on this particular enzyme. Okay. So, let us look at overall what we have done in case of the vanadium story is we have talked about the vanadium oxidation states, we have talked about the vanadium oxidation state which are important and prevalent in the biological systems like +5, +4, +3, and then kinds of vanadium species that we have been able to see, and we talked about the tools like vanadium 51 NMR is suitable and useful for vanadium 5 studies, and whereas the EPR is very highly suitable and useful for vanadium 4 kind of a species. We also shown that the vanadium 5 is not very stable when it enters inside the cell. They get further reduced to vanadium 4 and probably vanadium 3 in some cases too, for which certain specific cells, vanadobins and those kind of things were already, vanadochrome have already been explained and shown. Then we have shown that the vanadium vanadate species are dependent on the concentration of the pH. Then we have also shown the vanadate acts like a phosphate, and if it is present in the ATPase cycle during the phosphate removal, which is called dephosphorylation, at that stage if the vanadate is present, vanadate will bind to the protein irreversibly and then stops the cycle of the ATPase. That is the negative effect of the vanadium. Positive effect of the vanadium we have looked at through the haloperoxidases. Haloperoxidases are really a boon because they are the ones which are present in algae, in sea algae, and they are responsible in making the halogenated, brominated, chlorinated, iodinated products of the organic, which are very precious, very expensive, and which are very difficult to synthesize otherwise. Therefore, and then for such a kind of an enzyme we have looked at, in such an enzyme the vanadate species is suspended in the protein through one covalent link with one of the histidine and a large number of hydrogen bonds through the oxo species or hydroxo species present on the vanadium center. And all this acts in presence of the H2O2, and H2O2 reacts at this particular center, forms a peroxo species, which in turn is activated by the chloride and to form the hypohalous acid, and that is the hypohalous acid which is responsible for binding to the substrate, and that is being shown. And so, if the substrate is there, it will bind to that, and we have given the methods by which one can even assay the haloperoxidase activity of all this. So, we also shown that this particular thing is absolutely true and correct. The center, the vanadate, is the one which is responsible for haloperoxidase reactivity, because we could show by removing this and putting the sulphate, the enzyme is inhibited. So, therefore, these are the aspects. So, one inhibition of vanadate, one enzyme, and general features have been taught in this particular course. I think that information is more than less sufficient enough in this for vanadium story. And for the vanadium 3, instead of vanadium 3, I will take the molybdenum 3 for nitrogenase. So, in the story of molybdenum, I will explain the nitrogenase. So, I hope you understand what I am talking about, how these metal ions form, or metal ion species form, activate the enzymes, and then the reactions too. So, I think we are now geared to move to the next metal ion, and the next metal ion that I would like to take in the biological systems or the bioinorganic systems is the manganese. As you can see, that also comes later to the vanadium. Okay, vanadium and then manganese. Okay, the manganese also you know very well kind of oxidation states. We know we have seen most of the oxidation state +2 to +7. Of course, some other chemically available oxidation states are also there too. So, you can get large number of variety of oxidation states. However, not all of them are seen, as you will see. Manganese 2, manganese 3 and manganese 4 are more or less seen most of the time, 2 and 3 and 3 and 4. Very rarely the 5 may be associated, but never 7 or never lower than 2, because lower than 2 or 2 reducive, more than 5 is highly oxidative, and you know biological systems never take up the extreme kind of a species. If it takes up, it will control it, and that is what we need to keep in mind. Now, for a while let us look at this particular table to give a glimpse about the manganese enzymes. And let me tell you, manganese enzymes are diverse. There are manganese enzymes where one manganese is present. There are manganese enzymes where 2 manganese ions are present. There are manganese enzymes where 4 manganese ions are present too. And let us take some popular examples among these: manganese superoxide dismutase, I will come to this in a while, manganese catalase, manganese extradiol dioxygenase, manganese ribonucleotide reductase, manganese containing oxalate oxidase, oxalate decarboxylase, photosystem in this too. So, what is manganese superoxide does? So superoxide does, it breaks down the superoxide radicals into O2 and H2O2. So, O2 is harmless, H2O2 is less harmful than this O2 minus. So, therefore, such H2O2 is taken up by another enzyme called catalase, and there are catalases even on manganese and of course on the iron too. So, but I will be explaining in the manganese case. So, therefore, I may not explain this class of enzyme in the iron so much. So, manganese catalase, and this will convert the hydrogen peroxide to oxygen and water. So, overall the reactive species of oxygen, which is the O2 minus, which is superoxide radicals and peroxides, these are disproportionated and dismuted to be less harmful oxygen and water kind of a species. Occurrence is given, you can always go through that. So, these the manganese superoxide dismutase is the case where one manganese is present, manganese catalase is the case where the two manganese are present. I will explain that. So, manganese extradiol dioxygenase, and we will not be looking into this details of this enzyme. Manganese ribonucleotide reductase, will not be looking into the details of this enzyme, but we look at the reactivity. The reason is that the details of this enzyme I will explain you when we come to the story of iron. Iron is also involved in ribonucleotide reductase, but however, I will tell the reaction or function that is happening in this case, in this story itself. Then the oxalate oxidase will not going to take up, oxalate decarboxylase will not going to take up. Photosystem 2, where the 4 manganese are present and the water is oxidized to oxygen, O2. So, this is present in plants, this is present in bacteria, algae, etc. So, in this case we will study superoxide dismutase enzyme, catalysis, manganese catalysis, enzyme catalysis, photosystem enzyme catalysis. These are the 3 cases that I am going to explain you under this banner of the manganese biological inorganic chemistry. Before we go into the enzymes, I have put a slightly different mode of approach now. Here I have shown 1, 2, 3, 4, 5 different kinds of enzymes. I have not shown the enzyme, but I have shown the metal ion or a manganese center. So, you see the manganese center in the SOD, there is only one manganese here. This is bound by 3 histidines etc., and this whole thing is protein. And this is photosystem, there are 4 manganese, the purple ones are 1, 2, 3 and 4, and there is 1 calcium. This is called a cluster, tetramanganese calcium cluster. Of course, more details will be explained later in this class itself, in this particular under the story of the manganese itself. Then look at the catalase enzyme, which disproportionates the hydrogen peroxide to water and O2, and this has 2 manganese centers, as you can see here. There is one little manganese over there, another little manganese here. This has a core and this has a core. I will explain this too and mechanism too. So, all the 3 enzymes I am going to explain their mechanistic aspects. Then we have one other enzyme, which is ribonucleotide reductase. I will give just the function, but I will not be giving the reaction, the mechanism of this, because I will explain under the iron story. Manganese dioxygenase, I will explain this under the copper case and the iron case, dioxygenases, copper dioxygenases. So, we will give away with this. So, out of these 5 reasonably well known enzymes, these 3 of the enzymes I will be handling in under the story of the manganese. Let us look at the kind of reactions that it do. You see the oxygen, if you add 1 electron, it will become O2 minus. This is what is called superoxide, and if you put 1 more electron and put 2 protons, it will become O2 2 minus, put 2 protons, H2O2. So, the superoxide, what it does? O2 to H2O2, and what catalyzes that? From H2O2 to H2O. So, the right side part, this is catalase, the left side part is the superoxide, okay, and these kind of a systems. So, the H2O2 then further breaking to OH minus and then H2O. Okay. So, you can use this, and in the peroxidase enzymes also you can go through this kind of a step. Manganese dioxygenase, I told you that I will explain this when I come to the copper, when I come to the iron. So, but just I will show the example here, okay, and this the O2 has got 2 oxygens, these 2 oxygens are added up over here. See that the 2 more additional oxygens have come here and these are coming from here. So, this is called dioxygenase. Whenever an enzyme adds 2 O parts to the substrate, it is called dioxygenase. So, you will understand this much more better when we come to the iron case, when we come to the copper case. The other enzyme is RNR, it is called ribonucleotide reductase. What is ribonucleotide reductase thing? Let me tell a little bit in this. You know, the systems have got RNA and DNA. RNA has got ribosyl and DNA has got deoxyribosyl, and to the body what is input? Ribose, never deoxyribose. So, if body has to synthesize DNA, it has to synthesize the deoxyribonucleotide. To make the deoxyribonucleotide, it has to make deoxyribose. So, that particular enzyme which does this deoxyribose. What is deoxyribose? Ribose has got a 2' hydroxyl, deoxyribose has 2' hydrogen. So, this is what the difference is. So, converting the finally converting the 2' hydroxy to hydrogen. So, this kind of a conversion is called the ribonucleotide reductase enzyme, and after making the ribonucleotide then only the deoxyribonucleotide, the other system of enzymes will make the DNA, which that is not a part of this particular story. So, first part of making a DNA is to make the deoxyribosyl moiety or deoxyribosyl nucleotide moiety. So, to make that, take ribonucleus nucleotide and make the deoxyribonucleotide. So, and the mechanism of this I will explain when we come to the story of iron and probably even the story of the cobalt too. So, let us see what all we have done in this particular class is I have explained you several aspects related to vanadium, and I have already made the conclusion of the vanadium. And now I talk to you about the manganese entry. Manganese got several oxidation states, but most oxidation states favored are +2, 3, 4 and very little towards 5. Never very highly oxidized, never very low oxidized ones, because they are either strongly oxidizing or strongly reducing. There are manganese enzymes with 1 manganese, 2 manganese and 4 manganese. I will be taking one example of each. One is superoxide dismutase, other is the catalase, the third one is photosystem, where the oxygen is evolved from the oxidation of the water center. So, the kind of reactions are shown over there, and the last reaction here you can see that it is in the carbon dioxide going to the carbohydrate and the water going to the oxygen, and I will be explaining the details of this as well. So, all these 3 cases I will be explaining in the next class. Thank you very much.